Hello everybody, welcome back to lecture 26. Today we're covering the Calvin cycle and the Pentose phosphate pathway, both of which are pretty cool if you ask me. So go ahead and get started. First going to cover Calvin cycle. We already talked about the light dependent reactions, right? What you should know is the main idea is that we have water going in and we put some light in and we get ATP and ADPH out. And then we also produce oxygen. And then the ATP and ADPH actually end up going into the Calvin cycle to make sugars. And we take CO2, we fixate it, and turn the carbons from CO2 into sugars, basically. How does this work? We've got our steps of this reaction of the Calvin cycle here, and you'll notice that first of all, I want to specify that the number of things that go in is not just 1 to 1. So notice how we have 3 things going in here, we have 3 things going in here, we get 6 out, we have 6 going in here, 6 coming out, 6 going in here, 6 coming out, 6 coming out here, 1 leaving here, and then 5, and 3 and 3. So these are numbers you have to keep in mind. So it's not quite a 1 to 1 ratio, and it gets a bit more complicated than that. Although the number of carbons is the same, right? So you'll notice here, so 3, a ribulose is a 5 carbon sugar, so there's 15 carbons here total. We add 3 here, right? So that's going to be 18 carbons, and there's 6 3 carbon sugars here, so that'd be 18 carbons, right? So the number of carbons is constant across the entire cycle, but it does become a bit weird because we're not dealing with 1 to 1 ratios. So we're going to start with the first step here. We're going to do carbon fixation, so CO2 fixation. Essentially what you're doing is we have our enzyme Rubisco, or Ruby as we call it. I believe there's a slide, yep, Ruby. And essentially what this enzyme does is it takes the CO2 and condenses it with a ribulose 1,5-bisphosphate. So basically combines it. And when we combine it, we also end up splitting it. So we combine it and basically have this sort of aldolase activity where we split it in half and we make 2 3-phosphoglycerates. And the way that this reaction works actually is it's not just a 1 to 1. We actually put in 3 of these ribulose 1,5-bisphosphates. Otherwise, you see that sometimes people write this as RUBP, right? Like they write it like this. So if you see this, same thing as ribulose 1,5-bisphosphate. What we do is we actually put 3 of these in, and then we get 3 of these in. We put 3 of these in, and we actually get 6 of these 3-phosphoglycerates out, which should make sense. Basically, all you're doing is you're taking these 5 carbon sugars, slapping an extra 1 carbon on each of them, so you have 18 carbons total, and then you're splitting them into 6 different pieces. So then you end up with 6 3-phosphoglycerates. Okay. And they show you the math here. How important is this to know? I wouldn't say it's super important to know. Oh, classic me when I delete my accidentally delete everything. There we go. Okay. Anyways, yeah. As I was saying, Rubisco is made of 16 different subunits, and it's basically a carboxylase and it's a carboxylase and oxygenase. So carboxylase makes sense, right? Carboxylase and oxygenase, it breaks apart. It's a pretty slow enzyme, so 3 per second is extremely slow. You can think that's your kcat, right? Your kcat is 3 per second. It's 3, which is ridiculously slow. So you can imagine your Vmax is not going to be very large. And it can be up to 50% of the leaf protein. And it's the most abundant protein on Earth, which is cool. In terms of what you should know about cofactors, you should know that it needs magnesium and it works best at a basic pH, so optimum pH 8. So you have 8 large subunits with catalytic sites and regulatory sites, and we have 8 smaller ones that enhance the catalytic activity of the large subunits. So you have 8 supporters and 8 main ones, basically. So now we're on to stage 2. So we covered stage 1. Stage 1 is this fixation of carbon. Stage 2 is more of this cycle process. So we have our 6 3-phosphoglycerates, and what we do with that is we're essentially taking it and turning it into glyceraldehyde 3-phosphate, which is actually, if you remember, what we're doing before, right, is we're doing glyceraldehyde in glycolysis, what we do is glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate to 3-phosphoglycerate. So we're actually go this way in glycolysis, but we're actually going the opposite direction in Calvin cycles. We're just going in the reverse. So, and it says that right here, right? We're doing the reverse of glycolysis. That's literally all we're doing. And it's the same enzymes too. So if you look here, from the 6 3-phosphoglycerates, we do phosphoglycerate kinase to add the phosphate back onto this 3-phosphoglycerate to make 1,3-bisphosphoglycerate. Remember, because we have 6 of these, we're using 6 ATP, it's producing 6 ADPs. We make 6 of these 1,3-bisphosphoglycerates. And then what we're doing is we're going to reduce this into the glyceraldehyde 3-phosphate. Because remember, a carboxylic acid, like 1,3-bisphosphoglycerate, is more oxidized than glyceraldehyde 3-phosphate. So we put 6 NADPHs in, get 6 NADP pluses out, and 6 PIs, because we're getting rid of these phosphates, right? We're removing a phosphate and we're reducing it down into 6 glyceraldehyde 3-phosphates. And what ends up happening here is that one of these glyceraldehyde 3-phosphates leaves to go help become a sugar, right? And the other 5 go back and turn into RUBP, ribulose 5-bisphosphate. And the other thing is, where do we get these NADPHs from? Well, we get these NADPHs from electron transport chain, right? And the light-dependent reactions. Same with this ATP, right? We get this ATP from the light-dependent reactions. So if you go back up here, we're getting this and this from the light-dependent reactions and using them in these steps. And we already talked about this, right? It's notice you can look at this more if you want to, but it is just the reverse of glycolysis, right? And here we have the final step, which is the reproduction of RUBP. And what's happening here is, after we have our 6 glyceraldehyde 3-phosphates, and like I said, one of them leaves, right? So one of the 3 carbon sugars leaves, the other 5 go here. So we have 15 carbons in total. And so what happens now is we take these 15 carbons, we slap these some of them together, right? And then we phosphorylate them with ATP. So we take 3 ATPs. So what we're doing is we're combining the 5 3-carbon sugars that we have left. What is going on? What we're doing is we're taking these 5 3-carbon sugars that we have, we are slapping them together and producing 3 5-carbon sugars. 3 of these ribose 5-phosphates. And then we attach phosphates to them and we turn them into ribulose 1,5-bisphosphate. And the important thing to recognize about this step here in particular is this step is not that simple. This step, it seems very easy and not very difficult, but it's actually very complicated, what goes on in this step. And I'll kind of showcase that below, but I wouldn't expect you guys to memorize every single one of those intermediates. It's probably helpful to know them, but I wouldn't say that you need to memorize everything. What you should memorize is transketolases. So what does a transketolase do? That's very important. It's important in the regeneration phase. So important in this phase where we're regenerating our RUBP, right? So our RUBP is ribulose 1,5-bisphosphate, remember? So what is essentially happening is a transketolase will essentially take 2 carbons and transfer them from one place to another. And that is exactly what's happening here, right? So it takes 2 carbons and transfers them from one substrate to another. And the important cofactor for this is TPP, which is something that we have seen in pyruvate decarboxylase and pyruvate dehydrogenase. And generally speaking, what you should know about TPP is that 1, it is essential for decarboxylation reactions. That is the one thing. And 2, it is very important for transfer reactions. So in 441, you'll cover aminotransferases. They also use TPP. Essentially, It's just a very important vitamin or cofactor for the coenzyme, really, for these kinds of reactions that require transfer or decarboxylation reaction. And so here's a couple of other things that you should kind of keep in mind is that, from ribulose 5-phosphate, which is an aldose, right? Aldose, because it's got this, to turn it into, I think they flip these around. This is a ketose. This is an aldose. So flip these two. Yeah. So this is a ketose because it's got a ketone group here, right? There's a carbon here, and then this is an aldose because it's got an aldehyde on the end. So same molecule, right? Different form, different same molecule, so same molecular formula. Sorry, same molecular formula. Jeez, different chemical structures. So they're not the same molecule, but they are isomers of each other, right? So we've got the aldehyde form over here, and then the ketone form over here. And they can be isomerized, right, into each other with these different things. So phosphopentose isomerase and phosphopentose epimerase is a form of isomerase, but all the epimerase does is it changes one of the carbons, right? It just changes one of the carbon's chiral centers. It does not change anything else. This is different from isomerases because isomerases kind of like change the whole molecule around, but epimerases only change one chiral center. And here is the monstrosity that is the entirety of that process for regenerating RUBP. So to regenerate, so if you notice here, there's two different places where we regenerate ribulose 5 ribulose 1,5 bisphosphates, down here and here. And you'll notice that we generate 3 total, which makes sense because there are 3 total here, 3 here, and we generate 2 over here and 1 down here. And so what's going to happen is, if you have your glyceraldehyde 3-phosphate, that you have your 5 glyceraldehyde 3-phosphates, right, that are going through this pathway here. And remember, the glyceraldehyde 3-phosphate right here. You got glyceraldehyde 3-phosphate right here. You got glyceraldehyde 3-phosphate over here. And the other thing you should know is glyceraldehyde 3-phosphate can be turned into dihydroxyacetone phosphate using triose phosphate isomerase, or TIM, or whatever they call it. So we can make this, it's probably not good to use green there, use orange here, 1 and 2. And notice how many carbons are in these intermediates. We've got 1, 2, 3, 4, and 5. Notice how many 3-carbon sugars are produced here. It's 5. So it's 5 of them. So 2 of them are turned into dihydroxyacetone phosphate, these 2, and the other 3 are kept as G3Ps. You get the idea, right? So the 5 sugars, the 5 3-carbon sugars are just kind of changed and moved in terms of their carbon structures to make these things. So 2 of them become dihydroxyacetone phosphate, 3 of them stay as G3P, and then we do this massive carbon shuffle where we end up making 2 ribulose 1,5 bisphosphates and 1 ribulose 1,5 bisphosphate in this step down here. I can kind of go through what happens here on a very basic level. I'm not going to go in super in depth on what happens in each reaction, but I will show you the general gist of what's going on. So essentially, we're doing the reverse glycolysis in this reaction, right? We're taking our G3P, our DHAP, turning it into fructose 1,6 bisphosphate using aldolase. And then we use fructose 1,6 bisphosphatase to remove this phosphate, make this fructose 6-phosphate. This is a very exergonic step, if I'm not mistaken. Yep. Whole process, 3 exergonic reactions make this whole thing irreversible. Fructose 1,6 bisphosphatase, cytoheptulose 1,7 bisphosphatase, and ribulose 5-phosphate kinase. So this right here is a committed step, very negative in delta G. And then we have here fructose 6-phosphate with the transketolase. And transketolase does what? Transketolase, right, moves 2 carbons from one place to another. So notice we go from fructose 6-phosphate, which is a 6-carbon sugar, to erythrose 4-phosphate, which is a 4-carbon sugar. So we're actually moving 2 carbons from the fructose 6-phosphate to the glyceraldehyde 3-phosphate here to make xylulose 5-phosphate. We've made that. So we've done that with a transketolase. And then we just have to use ribulose 5-phosphate epimerase, because they're epimers, to make ribulose 5-phosphate. And this just goes into the ribulose 5-phosphate kinase to make ribulose 1,5-bisphosphate. Now, coming back to this erythrose 4-phosphate that we just made. Now what we're going to do is we're going to take our dihydroxyacetone phosphate. We're going to add an aldolase, right, our enzyme aldolase, to make cetohepulose 1,7-bisphosphate, which is a 7-carbon sugar. And from this 7-carbon sugar, what we can do is we can take another glyceraldehyde 3-phosphate with the transketolase, and then we make from that a ribose 5-phosphate and a xylulose 5-phosphate. So notice how we have a 7 and a 3 carbon, and we make 2 5s out of that. And then from there, for this ribose 5-phosphate here, we do ribose 5-phosphate isomerase to make ribulose 5-phosphate. Then we do ribulose 5-phosphate kinase to make ribulose 1,5-bisphosphate. And for this xylulose 5-phosphate, what you do is you do the epimerase again to make ribulose 5-phosphate, and then you use ribulose 5-phosphate kinase to make ribulose 1,5-bisphosphate. A lot of stuff there. I don't think you have to memorize every single one of these, but you should generally know what transketolases do. You should know that they transfer 2 carbon sugars. And you should also generally know, I would say, the amount of carbons that are kind of being shuffled around. So for this cetohepulose here, you should kind of have the idea that if you have a 7 and a 3, you're going to make a 5 and a 5. And then if you have this, let's see here, this 6 and this 3, you're going to make a 4 and a 5. That's stuff that you should know. So you should always be trying to make 5 as one of a 5-carbon sugar as one of your products, essentially. And we talked about here on this side, if you look over here, there are 2 other steps that are like very exergonic that drive this. It's the ribulose 5-phosphate, which is ribulose 5-phosphate kinase. They don't actually show it here, but it would be this step here. These 2 steps here are both ribulose 5-phosphate kinases. And if you look at cetohepulose 1,7-bisphosphatase is going to be this step right here. So this step is important. This step is important in terms of that, and then these 2 steps here are important because they are very exergonic. So I've kind of already talked about this before, but essentially, the synthesis of this extra triose phosphate, so this this one triose phosphate that gets released out into the universe to become a sugar, right? This one here requires, where is the full cycle? It requires 6 ATP here, 3 ATP here, and 6 NADPH. And 3 CO2 up here. That's what you need to know. It requires 3 CO2, requires 9 ATP, requires 6 NADPH, produces 9 ADP, 6 NADP plus, and 1 triose phosphate that goes off into the wilderness. Yeah. Regulation of the Calvin cycle. So it's basically a light-driven reduction of these disulfide bonds that causes the activation of the enzymes. So the light leads to a reduction, which leads to activation of enzymes. And the enzymes that are activated by light are these guys. So you can see that, these 3 here are in the carbon shuffle, and the glyceraldehyde 3-phosphate dehydrogenase is just within Calvin cycle itself in that kind of secondary phase. So these enzymes are inactive in the dark, so that, you know, the synthesis of this hexose does not compete with glycolysis, right? Because we don't need to synthesize sugars if we're struggling to make sugars to keep ourselves alive. And, you know, they talk about the blue ones that are specific to plants, and that should be kind of obvious. We covered these in glycolysis, and glycolysis obviously occurs in humans, so. Here's an example of how these things are activated. So ferredoxin is actually one of the ferredoxin is actually the thing that kind of transfers these electrons. So you have the reduced form of ferredoxin, the oxide form of ferredoxin, and that through thyroredoxin reductase, which is an enzyme, reduces this thyroredoxin, and this thyroredoxin goes in and reduces a bunch of other enzymes, and that's how it activates them. So you can think of it like this. Imagine that we have our glyceraldehyde 3-phosphate dehydrogenase. If we're in light, we're going to make a lot of this reduced form of ferredoxin. This is going to go ahead and reduce the thyroredoxin with thyroredoxin reductase. Then this thyroredoxin is going to go and reduce our glyceraldehyde 3-phosphate dehydrogenase to make it into its active form. So high light leads to higher thyroredoxin using thyroredoxin reductase, which leads to higher enzyme activity because these enzymes are active when these disulfide bonds are reduced. I'm not too sure why they decided to include these, but you can think of this like this. So in the stroma, right, that's where all of these, the Calvin cycle occurs in the stroma of the, or actually in the thylakoid, actually occurs in the lumen. But essentially what's happening is, once you make your fructose, right, fructose 6-phosphate shows up where it shows up, right here. What you can also do is you can use this to make starch. And so what happens is it occurs in the stroma of the chloroplast. So when you get your fructose 6-phosphate from your kind of carbon shuffle, you can isomerize it into glucose 6-phosphate. Mutase, so mutase is like an isomerase, but it just changes a specific group from one position to another. So we're going from glucose 6-phosphate to 1-phosphate, just changing one group's position. And then from there, we basically make this kind of ATP glucose intermediate, similar to how glycogen synthesis works, right? Where you take UDP glucose, now you're doing ADP glucose. And this is now used to make starch, right? With starch synthase. Instead of glycogen synthase, you have starch synthase. Very similar process though, right? Glucose 1-phosphate, use ATP to activate it. We hydrolyze this pyrophosphate to make this a very negative reaction that's actually forward going. And we use starch synthase to make amylase. And then we also have sucrose synthesis that occurs in the cytoplasm. So this is in the stroma of the chloroplast. This is in the cytoplasm of the cell, generally speaking. And what we do here is we have our glyceraldehyde 3-phosphate that comes from the stroma of the chloroplast that, you know, we made from that that extra 3 carbon sugar that's kind of shut off in the Calvin cycle is here now. It's transferred into the stroma of the chloroplast because all those reactions occur in the lumen, right? And then once it's there, it gets transferred into the cytoplasm. Some of it will get turned into dihydroxyacetone phosphate through triose phosphate isomerase. We aldolase it, turn it into fructose 1,6-bisphosphate, go through this pathway to make G1P. Then we do UTP, and we do UDP glucose. And then we take UDP glucose, combine it with a fructose 6-phosphate to make sucrose. Because sucrose is just a combination, right? Sucrose is just a combination of fructose and glucose. And so once you combine these two, you make sucrose phosphate, and then you make sucrose. I don't know. These two I don't think are that important. I think you keep them in mind, but I think there's other things that you can be tested on that are not this, not this, and this is not big of a deal. So we've already talked about these. We got the small and the large subunits. The large subunits are the ones that have like the enzyme activity, so this point here, and then the small ones are like supporters, like the hype men. And now we're on to the pentose phosphate pathway. So the pentose phosphate pathway is a separate thing from the Calvin cycle. I want to make that very clear. It's very separate from pentose phosphate pathway. And similar to the pentose phosphate pathway, it also produces NADP. Similar to the Calvin cycle, and the electron transport chain within plants, it produces NADPH, and it also produces riboses and other pentoses, but the usage is a little bit different. So for pentose phosphate pathway, it produces this reducing power to counter the damage of free radicals. So it's supposed to counter oxidative stress. And then the other thing is that it produces riboses, and these riboses and pentoses are used to make things like RNA and DNA. And they're also used to make things like NAD and FADH2, NADH and FADH2, and coenzyme A. These riboses are used to make these structures that we're so familiar with these days. And here's an example of that. So this ribose right here, this sugar, comes from pentose phosphate pathway. And it's also called the hexose monophosphate shunt. This is something you'll see if you take the MCAT. It might show up, but not that big of a deal. Okay, so when it comes to the pentose phosphate pathway, we have two main pathways. We have the oxidative pathway and the non-oxidative pathway. And the oxidative pathway is irreversible, and the non-oxidative pathway is reversible. Cool. And you should be able to look at me and say, this is irreversible because we are releasing CO2. Once you release CO2 as a gas, you are not getting it back. It's gone. And the other thing you should notice is that we also make NADPH, and this NADPH is used to make all sorts of things like fatty acids, glutathione reduction. So glutathione is the oxidative stress guy. It kind of stops the cell from being oxidized a bunch and then dying. And then it also helps with other detox things. Basically, it's a very, very, very helpful for keeping the cell safe and not exploding. The other thing we also make is ribose 5-phosphate, which is used in nucleotide biosynthesis, which you will learn about in 441. Let's start by looking at the oxidative phase, and you should have all of these structures and enzymes memorized, plus their intermediates. This is very important. For the oxidative phase of the pentose phosphate pathway, what we're doing is we're taking glucose 6-phosphate. So we're taking our intermediate from glycolysis. The glycolysis here, we're taking this intermediate, and what we're doing is if the cell, right, has already done a bunch of glycolysis and doesn't need to do more, it can just use the glucose 6-phosphate to instead do pentose phosphate pathway to make ribose 5-phosphate and NADPH. So these reactions are irreversible. They do not go in the other direction. What we're doing is we're first taking this glucose 6-phosphate and we are oxidizing it. See this ketone here now? We've got a lactone, is what we call these. Lactones are these things that have these esters. These ring esters are lactones. So we're using our NADP plus to make an NADPH, using glucose 6-phosphate dehydrogenase to make 6-phosphoglucanolactone. And it's a lactone because it is a ring. It is a ring sugar that has this ester group. That is a lactone. The 6-phosphoglucanolactone. Then we have 6-phosphoglucanolactonase. This is a hydrolysis enzyme that breaks open the ring. So basically what you're doing is you're taking this water, you're attacking here, and kicking this off to make, and this is that group, right? And so we're using 6-phosphoglucanolactonase to cleave this bond to make 6-phosphoglucanate. And then from here, we're using another NADPH, we're using an NADP plus to again oxidize this to make ribulose 5-phosphate. This is not ribulose 5-bisphosphate. That's RUBP. This is different. This is ribulose 5-phosphate. Before it was ribulose 1,5-bisphosphate. This is ribulose 5-bisphosphate. Ribulose 1,5-bisphosphate is in plants. This is in both technically, but this is in the pentose phosphate pathway. Ribulose 5-phosphate is not the same as ribulose 1,5-bisphosphate. So we use NADP plus with the enzyme 6-phosphoglucanate dehydrogenase to make NADPH, and we also release this CO2. So we go from a 6-carbon sugar to a 5-carbon sugar as well. You can think of it like this. These electrons come off, these go there, and then this that basically releases, and this goes ahead and grabs this H here, and these electrons come down. You can think of it like that. That's one way of thinking about it. And so these enzymes are pretty easy to understand, actually, because they just kind of use the name of the previous thing. So glucose 6-phosphate, right, uses glucose 6-phosphate dehydrogenase. 6-phosphoglucanolactone uses 6-phosphoglucanolactonase. And 6-phosphoglucanate uses 6-phosphoglucanate dehydrogenase. And generally speaking, you need to know, not only do you need to know what these things do, but you should also know, generally speaking, what the enzyme type is. This is an oxidation, so hydrolysis. This is an oxidation and a decarboxylation. So moving on to the non-oxidative phase. This is after you've made your ribulose 5-phosphates from here. What we're going to do is now we're going to take these 3 5-carbons, which is 15 carbons, and we're going to turn them into 12 carbons here and a 1 3-carbon. It's a series of 3 rearrangements, and these are reversible now. So we've been the non-oxidative phase is reversible, and so we're transferring either 2 carbons or 3 carbons. So transketolases transfer 2 carbons, and transaldolases transfer 3 carbons. So transaldolases were not in the Calvin cycle, but these are now different. And these 2 carbons, the transketolases, they use TPP, right? The aldolases do not. You know, big red here, reversible. Remember that. So, what we're doing is we're just going to do a bunch of carbon shuffles again. But this is just an example of what is happening in a transaldolase reaction. So here's an example of that. So in so if we look here, it cleaves the ketose and transfers 3 carbon fragments to an aldose. It's called a transaldolase because it transfers 3 carbons to an aldolase, an aldol sugar, or an aldose sugar, not an aldolase, aldose sugar. So we have a glyceraldehyde 3-phosphate. This is an aldose. We're taking in 3 carbons from this cytohapulose, this ketose, and we're transferring them here. And so we end up with, we go from a 7 and a 3 to a 4 carbon and a 6 carbon, a 4 carbon and a 6 carbon, fructose 6-phosphate and erythrose 4-phosphate. That's what transaldolases do. These do not use TPP. Do not use TPP, right? Different from transketolases, those use TPP. Looking at the non-oxidative phase of the pentose phosphate pathway, we have another carbon shuffle, essentially. This one's a little bit more simple, I think, to grasp. But from the... You're going to make your ribulose 5-phosphate from pentose phosphate pathway. And then what you're going to do is we're going to isomerize this. We're going to isomerize one of them, or a couple of them, and we're going to epimerase some of them. So we're going to epimerase them into xylulose 5-phosphate, and isomerize them into ribose 5-phosphate. From here, we're going to use it. We're going to have our. We're going to do a transketolase reaction. And we have our 5 carbon. And I'm trying to see how they're. Oh yeah, I see what they're doing. Yeah, it's a bit weird how they're doing this. The heck is going? Hold on a minute. I'm just realizing how they're doing like a bunch of weird stuff with this. I always get confused when I looked at this. I looked at this earlier and I still got confused. Let's see here. So essentially, we've got our ribulose 5-phos. We got our transketolase. 5 and a 5. Oh, yeah. Okay, I see. Okay, so, gosh, this is a ridiculous slide. So we got our 5 carbon, our 5 carbon. Or 2 5 carbons, right? And what you're going to do is we're going to do our transketolase reaction. So we're 5 and 5. Our transketolase transfers 2 carbons. So we're going to take 2 carbons from this, and we're going to transfer them to this ribose 5-phosphate here. So we're basically doing that. We're doing this. We're going here and here. And then we make acetoheptulose 7-phosphate by combining these two, and we also make glyceraldehyde 3-phosphate. We make a gap. So one gap is made here. From here, we take our, and I hope that makes sense, but basically all we're doing is we're transferring these two carbons onto this guy right here to make acetoheptulose 7-phosphate. From there, we also have our glyceraldehyde 3-phosphate. And now we have, since we have a ketose here and we have an aldose, we're going to transfer 3 carbons onto this guy. So we're going to do a transaldolase reaction, and we're going to transfer 3 carbons from this onto the glyceraldehyde 3-phosphate to make fructose 6-phosphate, which is a product in glycolysis as well, and erythrose 4-phosphate. So the options we have are that this fructose 6-phosphate could just go into glycolysis. The other option we have is now we also have the option to do a transketolase reaction with this erythrose 4-phosphate and this xylulose 5-phosphate, because remember, this is a constant cycle. So this fructose 6-phosphate goes off and into the glycolysis, and from here we take a xylulose 5-phosphate. So now we're going here, and an erythrose 4-phosphate. These are 9 carbons in total to make a 6-carbon sugar, fructose 6-phosphate, and a 3-carbon glyceraldehyde 3-phosphate. These both go into glycolysis. So we have a fructose 6-phosphate, an F6P, an F6P, and a G3P. That all go into glycolysis, essentially. And this is basically what happens if the cell needs certain things. So, if the cell needs ribose 5-phosphate more than NADPH, so it does not need NADPH, we just won't do the oxidative form. So remember, the oxidative form would go this way, right? You'd have a branch coming off this way to make NADPH, and then we'd have the 6-phosphogluconolactone and all that stuff. But we don't need to do this, so we don't do that. Instead, what we do is we go down to fructose 6-phosphate, and we go ahead and make ribose 5-phosphate. And same thing here. You can also use glyceraldehyde 3-phosphate to make ribose 5-phosphate, because remember, these are all reversible. Non-oxidative phase, it's all reversible, not irreversible. It's all reversible. Reversible. So it's all reversible. So we can just go ahead and take these glyceraldehyde 3-phosphates and these fructose 6-phosphates and turn them into ribose 5-phosphates. If we need both ribose 5-phosphate and NADPH, then we can just do the oxidative form. We just do the oxidative version of this. And we make NADPH, and we also make ribose 5-phosphate. Great. If we need more NADPH than ribose, then what we can do is we can make the NADPHs using the oxidative phase, and then since we don't need these riboses to make sugars, we can just put those into the carbon shuffle and make fructose 6-phosphate and G3P, and then we're done, and we just go through glycolysis. And then mode 4 is if you need NADPH and everything else. And I guess the other thing you should note from this, sorry, I should specify this. You can go, so it's not actually going to glycolysis here, it's going gluconeogenesis. So we're making glyceraldehyde 3-phosphate, and then these are all reversible steps. We can go up this direction, right? And we can keep doing this cycle over and over again. So we can basically just do this. Wee woo, and we keep going this way. Right? Same thing here, right? It's not going all the way down to pyruvate. It's going here, and then going up this way to make ribose 5-phosphate. Hopefully that makes sense. I apologize for that confusion. Essentially, if you need more NADPH and not riboses, You're going to do this whole process, right? Make these riboses, and then you're going to turn them into glyceraldehyde 3-phosphate, and then it's going to go back up this direction to make more NADPHs. And the last one is if you just want to do NADPHs and pyruvate. If you need both of these, then you just do it like normal. You do glucose 6-phosphate into ribulose 5-phosphate. You make NADPHs. You make these ribulose 5-phosphates. You make glyceraldehyde 3-phosphate, and you go the rest of the way through glycolysis. And the thing that regulates NADPH, that this production of NADPH, is NADPH. So if you make a bunch of NADPH, what happens is that it goes back and inhibits glucose 6-phosphate dehydrogenase, which is the first step in the pentose phosphate pathway. If you make a lot of NADPH, we're going to inhibit this G6P production, and from there we can go ahead and make more ATP. Specifically, I should specify this is inhibiting the oxidative phase as well. The G6P dehydrogenase is... And here's kind of the weapon of why PBP is important for detoxification. It's important because glutathione. Glutathione is... it's a cysteine, basically. And it's the cell's weapon against these reactive oxygen species that are like oxidizing everything. So it protects from free radicals and peroxidases. And NADPH is generated in the pentose phosphate pathway, and it's used to regenerate this glutathione. So under oxidative conditions, what happens is that, if I'm not mistaken, I believe this glutathione, this SH, gets oxidized to another glutathione, and they just remain oxidized together. But what ends up happening if you have a lot of NADPH is that those disulfide bonds are reduced, and you can regenerate these glutathiones, and they can go back to doing their job. If you don't have any NADPH, though, these guys stay oxidized forever, basically, and you just don't have any protection against oxidative stress then. So, and you'll notice that people who have mutations that decrease the glucose 6-phosphate dehydrogenase activity, that's that first enzyme in pentose phosphate pathway that makes the NADPH, one of the steps that makes NADPH, they're more hypersensitive to oxidative stress. And this makes sense because if you're not making NADPH, you can't regenerate these glutathiones, and these glutathiones are very important for protecting against oxidative stress, stopping oxidation of other cellular elements. And so if you don't have any glutathione, you end up oxidizing everything else in the cell, and it ends up breaking your cell. So other thing is, I mean, this is kind of, we talked about this. So glucose 6-phosphate dehydrogenase deficiency, it's fatal in terms of high oxidative stress. We talked about this, right? It makes sense because if you don't have your NADPH, what's going to happen is that you can't make, you can't remake this glutathione, right? These glutathiones here. And if you can't make these glutathiones, this inhibition never happens, and these radicals are just able to go ahead and do all sorts of damage to the cell. And I don't think you guys have to know the exact mechanisms for how these things happen, but what you should know is that when the mitochondria respires and it uses O2, plus other drugs and all this other stuff, every now and then you'll produce this kind of superoxide radical, and this superoxide radical, it turns into hydrogen peroxide. Hydrogen peroxides are ridiculously reactive, and they turn into these radicals, and they do all sorts of problems. You use hydrogen peroxide in chemistry, right? If you remember from organic chemistry, peroxides are radical reacting kind of a species, right? Or they're radical reacting chemicals. And so what you end up doing is, when you make these radicals, they kind of like bind to all sorts of things. They mess up protein structures, and they lead to all sorts of damage to DNA, proteins, lipids. And you need glutathione, because when you use glutathione, what you end up doing is glutathione inhibits, it basically stops these radicals from doing any of this protein damage. and other damage. That's the end of this lecture, though. I hope that was helpful. I think, like I said, I'm planning to do a review session on Monday. So if you have any questions, feel free to ask me. I'm going to be at the IC for the week. And I'm going to hopefully have that review session and post it that night for your exam. But yeah, hopefully this video was helpful. And I look forward to seeing you at some point, hopefully. And yeah, happy studying.